speaker is Professor Youn, Hong-Duk Youn, and he is actually a Seoul National University, and his talk is title is Metabolism-Linked Epigenetics for Determining ESC Differentiation. And please give a big hand for the Professor Youn.
Thank you, Professor Kim. And also, I appreciate organizing committee members for inviting me here. Today, I'm going to talk about metabolism-linked epigenetics for determining ESL differentiation. Especially, I would like to focus on the functional role of alpha-ketoglutarate, one of important metabolite, on the timing of ESL differentiation. Actually, the chromatin in our cell is composed of nucleosome, and where the DNA wrapped with the histone octamers. So if you take a close look at the sequence of this histone amino acid, you can find a lot of post-translational modifications, just like acetylation, methylation, and phosphorylation. But interestingly, this post-translational modification is deeply involved in metabolism because this PTM, there's a lot of many metabolites is utilized as substrate for this PTM. For example, the lysine acetyltransferase can catalyze acetylation of histone using acetyl-CoA, and this OGT can catalyze oxygenation of histones using UDP-GLUNAC. Also, this lysine methyltransferase and DNA methyltransferase can methylate DNA and histones using SAM, which is one of amino acid derivatives. Also, this LSD1 demethylate histones using FAD as cofactor, and SERT protein also used NAD for the acetylation of histones. Also interestingly, this DNA hydroxylase, which is a DNA demethylase, and Jumanji domain containing lysine demethylase, then utilize this alpha-ketoglutarate for demethylation of histone and DNA, which means that this metabolism is deeply involved in the epigenetic state of chromatins. The stem cell has very unique metabolism, but usually this terminally differentiated fibroblast cell utilize the mitochondrial oxidations for ATP production. But on the other hand, some pluripotent stem cells, such as iPS cells and the embryonic stem cells, has very high glycolysis activities. And then this mitochondrial activity is weakened in stem cells. In our lab also, we show that core pluripotent factors such as OCT4 and SOX2 can target glycolytic enzymes. The hexokinase 2, which is the first enzyme of glycolysis, and PKM2, which is the last enzyme of glycolysis. And then this OCT4 can increase the glycolytic flux in embryonic stem cells, which affect differentiation of stem cells. This group also shows that they found that the embryonic stem cell has very strong glycolysis activity, and then through this glycolysis pathway, this stem cell can make a lot of acetyl-CoA, and then this acetyl-CoA take part in the histone acetylations, which makes active chromatins in stem cells. Also, they using the glycolysis inhibitor, they found that this acetyl-CoA production is a very important early differentiation of embryonic stem cells, which means that this acetyl-CoA production is very important for histone acetylations on chromatins. In 2009, this group screened which amino acid affect the stem cell pluripotency, and then they found that if you deleted the threonine in the medium, the stem cell completely lost the ESL pluripotency, which means that threonine is very critical for mouse embryonic stem cell pluripotency. And then later, through the another science paper, they found that threonine dehydrogenase, which is the very first enzyme of this threonine degradation pathway, this TDH is the OCT4 SOX2 nanog target genes. Actually, this figure shows that the threonine degradation pathway, and then through these very complicated pathways, actually this threonine is converted to SAM, which is amino acid derivatives. And then SAM is take part in the methylation of histones and DNA methylation. But in case of humans, human does not have an active TDH. Instead, the human provide the SAM from the methionine, the other amino acid. And then SAM take part in also the same methylation of histones and DNAs. In humans also, if you knock out this kind of enzymes involved in this SAM production, also human stem cell lost pluripotency, which means that some kind of amino acid also very important for epigenetic state in embryonic stem cells. Alpha-ketoglutarate is also very important metabolite for DNA and histone demethylations. In 2014, the Johns Hopkins group published that intracellular alpha-ketoglutarate maintains the pluripotency of embryonic stem cells. Also, in the middle of our working, they published these papers. But we still, at that time, so we still don't know which metabolism or enzyme governs alpha-ketoglutarate production in embryonic stem cells. Also, we still don't know how the alpha-ketoglutarate fine-tunes the fate of embryonic stem cells. And then this fact prompts us to keep on this working. So first of all, using the publicly available RNA sequencing data, we screened most of alpha-ketoglutarate producing enzymes. And then you can see here that PSET1 is highly expressed in embryonic stem cells. But once cell is differentiated, their expression is decreased. The pattern is very similar to core transcription factors such as OCT4, NANOG, and SOX2. Also, using the publicly available CHIP sequencing data, we found that OCT4 and SOX2 nanog target PSET1 in embryonic stem cells. Also, we can find that the OCT4 SOX2 binding motif in the promoter of PSET1. So you can see here that OCT4 and SOX2 nanog is very strongly binds to the gene bodies of the PSET1. And then using this CHIP experiment, by a CHIP experiment, we also found that OCT4 and SOX2 nanog very strongly binds to the gene bodies of PSET1 here. But once, if you remove leaf from the medium, this OCT4 and SOX2 nanog binding is significantly reduced. And then from this data, we can say that PSET1 is OCT4 SOX2 nanog target genes and highly expressed alpha-ketoglutarate producing enzymes in mouse embryonic stem cells. And then what is the PSET1? Actually, PSET1 is phosphoserine aminotransferase 1. It's one of the components of serine biosynthetic pathway. So you can see here that these three enzymes is component of serine biosynthetic pathways. So you can see here that PSET1 can convert to glutamate to alpha-ketoglutarate by catalyzing 3-phosphahydroxypyruvate to phosphoserine. this PSET1 can produce alpha ketoglutarate. So you can see here that OCT4 SOX2 nano is strongly binds to gene bodies of PSET1, but this kind of co-transcription factors cannot bind to PHDDH, the first enzyme of this pathway, and PSPH, the last enzyme of this pathway. That means that PSET1 is the only one regulated by OCT4 and SOX2. And then you can see here that the PSET1 is highly expressed in embryonic stem cells. But if you remove leaf from the medium, the PSET1 expression is gradually decreased. And then after 4 days, the expression is reappeared and then gone. But the reason why the PSET1 is reappeared after 4 days is that ATF4 is another transcription factor which can regulate the PSET1s. So you can see here ATF4 is also expressed in after 4 days. That's why PSET1 is rebound after 4 days. But anyway, usually PSET1 is gradually decreased during the ESL differentiation. And then using ZHBT-C4, which is a genetically engineered embryonic stem cells, so if you treat the doxycycline, the OCT4 expression is gone. At the same conditions, PSET1 expression is disappeared. But if you back up this exogenous OCT4 here, the PSET1 expression is rescued. That means PSET1 expression is dependent on OCT4 expressions. Also, using another genetically engineered embryonic stem cell, 2TSS22C cell. So here, if you treat the doxycycline, this SOX2 expression is disappeared. And then also, you can see here that PSET1 expression a little bit decreased, and when you back up this exogenous SOX2 here, the PSET1 expression is rescued. That means that also this PSET1 expression is dependent on SOX2 expressions. So here, we can say that the PSET1 is highly expressed in embryonic stem cells and then rescued by OCT4 and SOX2. And then we knock down this PSET1 in mouse embryonic stem cells. So you can see here, if you knock down this PSET1, the expressions of OCT4 and SOX2 nano KRF4 ESRRB is reduced. So also you can see here that if you knock down this PSET1, this stem cell lost their pluripotency by AP staining. And then if you back up this flag-tagged human PSET1, so you can see the expression of exogenous human PSET1, and then the expressions of OCT4 and SOX2, in the other thing, also rescued. That means that if PSET1 can maintain the expression of OCT4 in embryonic stem cells. So you can see here, so if you back up this flag-tagged human PSET1, this ES cell looks like wild type, even though it's not perfect. So if you compare this PSET1 knockdown cell, you can see that this cell is much better than this knockdown cell, which means that this PSET1 rescued the ES cell pluripotency. And previously, I told you that PSET1 is alpha ketoglutarate producing enzyme. And then we measured intracellular alpha ketoglutarate levels in PSET1 knockdown cells using the mass spectrophotometry. So you can see here that if you knock down the PSET1, the intracellular alpha ketoglutarate level is reduced. But if you back up this human PSET1, the alpha ketoglutarate production is increased. That means that the PSET1 is indeed regulating intracellular alpha ketoglutarate in mouse embryonic stem cells. And then we knocked down 3 different enzymes involved in this serine biosynthetic pathway. And then we detected the intracellular level of alpha-ketoglutarate. So you can see here that if you knock down this PSET1, the alpha-ketoglutarate level is most significantly reduced compared to this wild type. And second one is PHDH. Actually, the PHDH is the first enzyme. So if you knock down this one, so actually PSET1 cannot efficiently catalyze this one because PHDH1 cannot supply this substrate for PSET1. But if you knock down this PSPH, the alpha-ketoglutarate production is not much different between this wild type. This means that this PSET1 is a really affect the alpha-ketoglutarate production in mouse embryonic stem cells. And then we try to supplement of dimethyl alpha-ketoglutarate because actually this alpha-ketoglutarate is not cell permeable, membrane permeable. And then we used dimethyl alpha-ketoglutarate, which is a cell membrane permeable alpha-ketoglutarate derivative. And then we treated this dimethyl alpha-ketoglutarate in PSET1 knockdown cells. So you can see here that if you treat this alpha-ketoglutarate derivative, the expressions of OCT4 SOX2 nano-KERA4 is rebound. I mean, the expressions are high compared with this knockdown. Also, if you look at this stem cell morphology, this AP staining is better than this knockdown cells when you treated dimethyl alpha-ketoglutarate, which means that even though without PSET1, so if you supply the exogenous alpha-ketoglutarate, stem cell may stain the embryonic pluripotency. And then actually this alpha-ketoglutarate is very important substrate for DNA hydroxylations by TET enzymes. And then we purified the DNA from this PSET1 knockdown cells and the flag human PSET1 backup cells. And then spotted this DNA by twofold serial dilutions and then Western blot with 5-hydroxymethyl-cytosine antibody. So you can see here in PSET1 knockdown cells, the 5-hydroxymethyl-cytosine level is significantly reduced because if you knock down this PSET1, the alpha-ketoglutarate level is reduced, and then they cannot supply the substrate, cannot substrate for TET1 enzyme activity. That's why this 5-hydroxymethyl-cytosine level is reduced in here. But in this PSET1 backup cells, the 5-hydroxymethyl-cytosine level is very similar to wild type. Because I think this PSET1 can supply this alpha-ketoglutarate for these enzymatic reactions. Also, the publicly available, the 5-hydroxymethyl chip sequencing data, we confirm that this 5-hydroxymethyl-cytosine level is reduced in the gene bodies of OCT4 and SOX2 nano-KERA4 ESRRB using this chip experiment. So this means that the PSET1 can control the alpha-ketoglutarate levels, and then this alpha-ketoglutarate level is involved in the 5-hydroxymethyl-cytosine in embryonic stem cells. Also, this alpha-ketoglutarate is a substrate for Jumanji domain containing histone demethylase. And very similarly, if you knock down this PSET1, but in this case, very interestingly, only A3K9 trimethylation and A3K36 trimethylation is increased. This reaction is very specific. Also, the chip experiment, we found that this A3K9 trimethylation is increased in PSET1 knockdown cells. That means that also PSET1 can regulate histone-methylation state in embryonic stem cells, especially this A3K9 trimethylation and A3K36 trimethylation. And then we check up the alpha-ketoglutarate level during random ESL differentiation. So you can see here that once stem cell is differentiated, alpha-ketoglutarate level is gradually decreased and then rebound. This pattern is very similar to the PSET1 expressions. That means we believe that the PSET1 expression is really correlated with alpha-ketoglutarate productions during ESL differentiation. And then we treated this cell membrane permeable alpha-ketoglutarate in very 0 days after 1 day, 2 days, and 3 days upon leaf removals. And then we developed expressions of core transcription factors. So you can see here that if you treat this alpha-ketoglutarate in 0 days, the expression level of this core transcription factor is retarded compared to this wild type. And then after 1 day, also the expressions are retarded. But after 2 days, it's a little bit, I don't know, but in some cases, OCT4 is not much different, but SOX2 is a little bit retarded. There's a confusing state. But after 3 days, you cannot find any retard of expressions. That means that this alpha-ketoglutarate is very important for early differentiation of embryonic stem cells. Also, we compare these expressions of core transcription factors with the PSET1 knockdown cells. So you can see here that in this case, the expressions of these core transcription factors is shortened in PSET1 knockdown cells when compare these control ES cells. But if you supply this cell membrane permeable alpha-ketoglutarate in PSET1 knockdown cells, and then the expressions is retarded partially, which means that even though without the PSET1, just the metabolite alpha-ketoglutarate is very important for maintaining embryonic stem cells. And then finally, we try to make injected the PSET1 knockdown embryonic stem cells to skid mouse, and then we generated teratoma cells. So you can see here that the size of teratoma cells of this PSET1 knockdown is much smaller than wild type. Also, we found that these teratomas from PSET1 knockdown clones and differentiated improperly. Actually, you can find a lot of immature endoderms. Okay, from this data, we make a schematic model just like here. So actually, PSET1 is OCT4 SOX2 nano target genes, and then highly expressed in stem cells. And then this alpha-ketoglutarate utilizes substrate for this TET and histone demethylase, and make the very active epigenetic state in embryonic stem cells. But once stem cell is differentiated, PSET1 expression is gradually decreased. Also, the alpha-ketoglutarate level is decreased. And then in the early time stage, I guess that the active state and the repressive state is mixed up in maybe a transition state. But after 3 days, and then this 5-hydroxy-methyl cytosine is gone. Instead, methyl cytosine is appeared in here. Also, very repressive histone code markers, A3K9 trimethylation accumulate in here, and then make very repressive epigenetic state. So from this data, we suggest that this alpha ketoglutarate fluctuations during differentiation may determine the epigenetic state of stem cells and eventually determine the fate of embryonic stem cells. Actually, this work done, most of this work done by my very excellent graduate student, In-Young Hwang, now become the postdoc in my lab. And she's gonna move to Europe maybe very early in next years. Also, I express my thanks to all the members in my lab. Also, I very appreciate Professor Jung Do-hyun and Jeon Yoon-kyung for helping me analyzing immunohistochemical staining. And also, I appreciate Professor Park Sung-hyuk for helping me analyzing alpha ketoglutarate level using the mass spectrophotometry. Thank you for your attention.
Thank you. Thank you for nice talk.